So this video is on the scientific method, which is something that you've probably seen in other science classes and are relatively familiar with. So the scientific method, we often think of as a series of linear steps as to how to do science. And if you remember from kind of the last module, the science is really a way of knowing, and the scientific method is one way of knowing. It's the process we go through to which we acquire knowledge. And you've probably always learned this as a very linear process in which you start with an observation, you go down to researching your observation, making hypotheses, conducting an experiment, collecting data and analyzing that data, and finally drawing a conclusion which either supports or refutes our hypothesis. And this is a very standard scientific method. What I want to do is talk about kind of each of these pieces in the scientific method and then explain why thinking of the scientific method as a linear process isn't really the correct way to think about it. So the scientific method actually always begins with observation. As scientists, we're always looking at the world, trying to figure out how things are going on, what's causing these processes. So keen observation is how science really always begins. In this case, I put a picture up of a plant that you're probably familiar with from your biology course. It's a plant called Elodea. And the observation that I'm making in this case is that when placed in the water, Elodea is giving off some bubbles. So that's a pretty keen observation. In this case, because we see the bubbles, we're making observations because observations are things you can physically see or measure. The other thing that we make in science besides observation is something called an inference. An inference is something you assume based on your observations. So in this case, I'm assuming these bubbles are oxygen gas. They could be carbon dioxide, they could be something else, we don't know. But I'm assuming they're oxygen because I know that Elodea is a plant, and I assume that it's undergoing photosynthesis. Therefore, my inference is that these are oxygen bubbles. So once we make an observation and have some inferences on that observation, we become curious about this. So why is this Elodea plant producing bubbles? The next step is to do some research. So you either go to the library and get a book, or you pull out your computer and search the internet, and you do some background research about the process that you're observing. In this case, the Elodea plant is creating bubbles. And from this observation and from this research, you're finally able to do the next step, which is to ask a question about the natural world and how it works. So now that you've done your research, you have a question. And in this case, I was looking at Elodea plants. I've decided that a very good question I want to ask is, how does the amount of light the plants are exposed to affect the amount of bubbles the plants are producing? So I've got this question. I wrote it down. How does the amount of light affect the amount of bubbles Elodea plants produce? And I'm going to go back to my research and do research on things like photosynthesis, how light affects photosynthesis, what Elodea makes when it produces bubbles. And I'm ready to go ahead and investigate this question through what you think really is a scientific method. So now that I have my question and now that I've done my research, I can make a hypothesis. And you're probably familiar with the fact that a hypothesis is an educated guess as to the solution to a question. So in this case, we're talking about Elodea, we're talking about bubbles. We're going to say that as the amount of light increases, the number of bubbles should also increase because photosynthesis is happening faster. The thing to remember about your hypothesis, though, is it must be based on the research you've already done. So you're not just making a wild guess, you're making an educated guess. It also has to be testable, so it's something we could do in an experiment. In this case, if we increase the amount of light, we can measure the bubbles and vice versa. So it's very testable, it's clear, it's measurable. I can measure bubbles either by counting or by putting an oxygen sensor up to check how much oxygen is being produced. And it's just straight to the point. You may have been taught in the past that hypotheses have to have if-then statements. This is not necessarily the case. Often they might say something like, as the amount of light is increased, the rate of photosynthesis increases. Or because if I increase the amount of light, then. So you do have those if-thens in there, but you don't have to have an if-then statement to be a hypothesis as long as you're able to predict the relationship between variables. As a side note, part of your MYP grade is to create hypotheses. To do your very best, not only do you have to make a hypothesis and support your hypothesis with research, you also have to make predictive values of your dependent variable based on how you're changing your manipulated or independent variable. But we'll talk about this later when we get to experimental design. So once you've made a hypothesis, the next step is to actually run the experiment. We're going to spend the entire next little subunit on experimental design and the parts of a valid experiment, but I want to mention that this is where experiment falls in the scientific process. Once you've conducted your experiment, you're going to have a whole bunch of data that you need to analyze. And the way we analyze data is we put it into tables or charts, we do some math to it, and then we graph it to look for patterns between our variables in the experiment. We're going to spend a little more time talking about the correct way to graph and the correct way to analyze and create data tables, but this is the part where we analyze our data to see what the experiment told us, if anything. The final part of the linear scientific method is to draw a conclusion. So you take your data, you see what your data told you, and then you figure out what you learned from this experiment. Any valid conclusion, particularly in a written lab report though, has three main parts. So the first thing you want to explain in conclusion is what you did. This is basically the outline of your procedures of the experiment and explaining what you were trying to investigate. The second part is what you learned in the experiment. So how did the amount of light affect the bubbles LED it produced? And you want to give the specific data points or the trends that you analyze that support this relationship that you learned. And finally, the third thing you want to explain is whether or not your hypothesis was supported. If your hypothesis was supported, great. If it wasn't, you may need to go back to the drawing board and redo the experiment. As a side note, with your hypothesis, if it was supported, it does not mean it was proven. It's very important to remember that we can never, ever prove anything in science. At the end of the day, though, the scientific method is not linear. Even though you start with observations and work your way through experimentation to eventually come to a conclusion, it doesn't necessarily mean that you're done. You may discover new things along the way in the experiment, new things to observe, new things to investigate, and start the process all over again. Or your hypothesis may be disproven. You can disprove things even though you can't prove things. And you need to come up with a different experiment to show that your hypothesis was supported. All in all, science is a never-ending process. We are always continually learning, continually finding new things, and continuing to investigate, refine, and redo our theories. That's why even though you think the scientific method is linear, it's not.